we're zooming in even further. We're going to look at proteins. I gave myself a tall order with the title that I chose, Understanding Life. I'll talk about protein interactions, right? And the understanding is future work. I'm working on it. I'm not there yet. This should work. Yes. So why are we looking at protein interactions? This is one of my favorite images, and I'll be moving a lot, so you have to be quick. So this is a synaptic bud. This is one of the small protrusions that you have in the synapse. And it's the sending part. So you see vesicles here that carry the neurotransmitters that are released. There's one somewhere that's in the process of releasing its cargo over here. And so this is like a snapshot of a molecular machine in action. This is fairly realistic, but it's not real. It's an artist impression. And so what they did is they measured all the proteins that they could measure with fluorescent labeling, with all sorts of other technologies. They also know the structure of, this was pre-AlphaFold 3, but they still had, most of those proteins were already, many of them were already solved in crystal structures. Yeah, this is a paper from 2014. So this looks like it's done, right? Like you could actually put this in a molecular model and you could run it and it would work. But there's a couple of things missing, but the first big thing that's missing is that although all the proteins are in there in the right quantities and roughly the right location, there's no knowledge about their interactions in this depiction. So if you would build a car like this, you have all the engine parts in the front of the car, unless unless it has it in the rear. You'll have four wheels around the car. You have the seat components and the steering wheel lying on top in the inside of the car. Everything is in the right place, except it's not. Right? This car will not go. This synapse will not go. It's a nice picture. It's great. It's missing all the interactions. So it's like an IKEA closet, but you have just bought it, right? Yeah. So the interactions are not correct here, right? Yet. Okay. So that's why we look at protein interactions, because this is the biology, right? So this is a connection to understanding life. Now, so it's about understanding function. Many disease-causing SNPs or other things happen on protein interfaces. So if you have a new virus, then understanding and most new viruses have a couple of new genes like that are unlike anything else we've seen. You want to have a quick view on what could these proteins be doing? Is there an interaction interface? Does it bind to another protein? Does it bind to DNA? But does it bind to small molecules? Every day might help in that case. Other pathogens as well. I made this slide in 2017, so I'm not on the hype. It was before the hype. This is interesting. I was going to go to the next slide, but this is definitely not the next slide. Right, yes, so. But before I go into the details, I want to tell you what protein interactions are. Forward. Okay. No, there's something weird with my mouse. Yes, so if you read papers about protein interactions, they can mean many things. So I want to disambiguate that. So interactions can mean indirect interactions like mutual dependence, where you measure some relation between A and B, but it's because when you remove C, they don't. So it's because they're both depending on C. Or it's a cascade where you measure a dependence on the level of A versus B, but A has nothing to do with B. A influences C, C influences B. How they influence each other could be many different things, but this is labeled as interactions in the literature. Then a little bit more to the physical side is complex membership. So this is what you get out of co-IP experiments, immunoprecipitation, where you basically extract complexes and you know that your bait protein and your target protein are in there, but they could be on opposite sides of the complex. They're never touching each other, yet they're measured as an interaction. So if you go to the string database, many of the interactions that are reported there are like this. There's nothing wrong with that. It's just that you need to know what you're talking about. So what I'm talking about when I say protein interactions, is I'm actually a physicist by heart. I was a molecular biologist by training, but I like the physics. I mean physical interaction, right? That kind of stuff. Okay, so that's the protein interaction. So so then what do we mean when I say protein interface residue prediction, which was in my previous title, but it's no longer in the title, so I'm saying it now. So we're predicting which residues in the protein are likely to be part of the interface. So if you have a crystal structure somewhere, and this is how these two proteins are in complex, then all the contacting residues, open it up, you can see them on both sides. Those residues is what we call the interface. And what we want to do is we want to do this sequence based. So we want to predict from sequence. So we basically have a sequence as input, and then we want to identify which are the yellow residues that are likely to be on the interface versus the blue ones that are likely not on the interface. And it's binary, so we say yes or no. Of course, like most prediction methods, it outputs a scale, so you can set a threshold, you can optimize that, you can benchmark this, you know the stuff, rock plots and so and everything. I probably have a rock plot somewhere in the slides. Okay. Yes, that's what I just said. And why sequence based now that we have AlphaFold? Well, not all proteins have a structure. For not all proteins that have an AlphaFold prediction, we know how good it is. We know how good it is for proteins that look like crystallized proteins, because that's what AlphaFold was trained on, and that's what we can benchmark it on. But we don't know how good AlphaFold is on proteins that are hard to crystallize, because we don't have crystal structures of them, or very few. Right? So we can benchmark it on the proteins that we know it's going to be good at, but we don't know how to benchmark it. Well, we do know with some ideas, but that's difficult. That's for another lecture. And we also have to still figure out what are the weak and the strong points of AlphaFold predicted structures. And the final thing is that if you want to do structure-based prediction like this with AlphaFold, you need to know the binding partner, not just the binding partner. You actually need to know the stoichiometry. Is it AAB or ABB, or is it AABC or just AB? If you don't know, you have a combinatorial explosion. You have to try all combinations and hope that something floats up. If you do it sequence-based, you don't have any of this complexity. You just input the sequence, you train your classifier, and you have an output. Now, right. So there are multiple ways to predict protein interactions. This is just a quick overview. The very fast and dirty methods, they use phylogeny, because if two proteins evolve in a similar way across many species, they're likely related, but this could be a very indirect relation. You could do docking, structure-based. It's very slow, it's very inaccurate. I would not recommend doing docking unless you have no other choice. You could do molecular dynamic simulations. They're far slower than docking, but they're more accurate. So you actually get cool results. And I'll show you a little bit in a minute. And they allow you to quantify the interaction strength, which is very important, because if you, for example, have 3 possible stoichiometries for your interaction, you can calculate the interaction strength for all 3. Then the one with the highest calculated affinity is probably, or is the most likely one to be correct. You never know. You have to benchmark that, rock plots, and so on. You know that. Okay. So, so from the top, it's knowledge-based, because you use all the sequence data that you can do for your phylogeny and so on. And at the bottom, it's first principles, because we use knowledge of how atoms interact and all the detailed ones. And this is the age-old spectrum of modeling. It's potentially very fast at the top. It's potentially very accurate at the bottom, and you have to find some middle way where the calculation is tractable and the output is useful for whatever you want to do. Okay, so this is what you get if you do molecular dynamic simulations on a complex of MP1 and P14, I have no idea anymore what, I wrote a paper on this some while ago. Don't remember what the proteins do. It's a nice one. It's a very small one. It's over here. You calculate the forces as a function of distance. You integrate the forces. You get the potential of mean force, which is an estimate of the free energy of binding. And what we did here is we did this with two force fields, a very detailed atomistic force field is very expensive, and a coarse-grained force field that's way simpler, about 1,000 times faster to simulate. And of course, the profile that you get, so the depth of this valley here with respect to the long distance when they're not interacting, that's your binding affinity. You can see they're different, but this is the experimental binding affinity at the distance that they are in the crystal structure. This is the expensive atomistic simulation. It's too strong binding, it's too short distance. So you can't say from this that the coarse grain is worse. I'm gonna say it's better. I don't know if that's statistically significant. Definitely not worse. So this works, you can do it, it's expensive. The all atom one takes about 2 months of calculation on 15 cores or 16 cores or so. This one is 1,000 times faster, so it takes only a day or 3. These are slow calculations, but they're cool and they can be very accurate. Now, you can do other stuff because if you have these fast simulations with coarse-grained force fields, you can do all mutations on the interface, and then you find that the difference in free energy when you make a mutation, the delta G, actually correlates with the Blossom score. This is a nice one. I like this one very much because this is a direct comparison of physics, biophysics with evolution. These simulations are on the order of nanoseconds, and Blossom 62 captures hundreds of millions of years of evolution, and there's a correlation of 0.4. I find this really awesome. Okay. The actual topic of my talk is machine learning, because everybody does it, and it's actually useful if you have good data. So we wrote a paper on this, how to do this. And generally, if you want to predict things from sequence for protein properties, could be protein interactions, could be folding, could be early folding sites, could be all sorts of things that you're interested in. Not anything, because not everything is equally easy to predict from sequence, but many things you can. And there's a couple of things that you need to get right, and we wrote this because we've seen, well, we made some of these mistakes ourselves, but we also saw a lot of these mistakes even in published papers, but certainly in manuscripts that were reviewed. So we thought it was good to have 10 quick tips for how to do this better. So we also did this. So this is in the wrong order. I want to have one slide in between. Okay, anyway. So for the protein interaction prediction, we also did a machine learning exercise. We have a data set of about a few thousand proteins with known interaction sites from the protein data bank, crystal structures of complexes of 2 proteins, or sometimes 1 protein twice when it's a homodimer or multimer. And whatever is close to the other protein is interacting, and you annotate your sequences. You have a training set, test set, and so on. We used PROPT5 embeddings, protein language model. This is just a picture from the Elnagar et al. 21 paper. It's a very famous paper by now. Just showing you some Disney projections in 2 dimensions of the embedding space to show that this model actually learned patterns from raw protein sequences, unannotated sequences, and then from that they extract a vector of embedding values of 1,024 values that you can then use as input for your machine learning model to predict whatever you want for the protein, but it already knows what structures of amino acid sequences you can find in real proteins. And that's a huge advantage that I'll show in a minute. So this is what our own architecture looks like. This is the previous version when we still used classical input features like well, the amino acid sequence, the PSSM from BLAST, predicted secondary structure, and some other things. And we fed that into 6 different machine learning architectures because we also wanted to know which one would work better. And it turns out they all had their weaknesses and strengths. So in the end, we fed all of their outputs into an ensembling network that's here in the middle. I can't look at the slide because there's a delay between when I'm moving my mouse and when the cursor moves. I have to look here. And that worked pretty well. But this is not using embeddings yet. So now we have an embeddings model. We went back to the architectures. We did a little bit of ablation to see whether all of these architectures were still having any added benefit for the prediction. And it found out that 3 of them actually don't do anything anymore when you have the embedding models, which is an interesting insight in itself. So we're down to 3 architectures in an ensemble. And then we can compare them. So this is area under the curve of the rock plot. For you in the master, I don't have to explain this. You can explain this to the people that don't know what it is in the coffee break. And so Pippin is the old model that used the handcrafted, old-fashioned input features. Pippin EMP is the same architectures, the 3 ones, and with the embedding input. And you can see it goes from something like 0.75 to 0.80. So clear improvement. But if you try it on different data sets, you get slightly different results. So this is also something you have to be aware of, that there's nothing absolute about performance of a method. It can depend on the data set. There's a different one that was published a while ago. And then there's another one that was published a week before we submitted our manuscript. And that is doing slightly better than us. But it's doing slightly better than us on a data set, a benchmark data set that was published in 2019. And if we try a different test set, like the test set that we were working with, that we published in our Pippin paper, not so long ago, then the performance difference went away. And then if we do a very small data set of Mycobacterium tuberculosis proteins, there was a difference again. So we didn't understand exactly what was happening there. For reference here, on that small data set, because it was otherwise too tedious to run, we did 2 structure-based methods, Pesto and CSM potential 2. And then you can see that you would think the structure-based prediction is better than sequence-based prediction. Well, these are very recent and state-of-the-art methods. And the difference is interestingly small. Let's put it that way, right? Okay, and there's also a difference between different structure-based methods. So it's not as easy as saying that it's always better to use a structure. Okay, this is what I was just telling to you. Yeah. So, and performance is different between data sets. So why is that? 2 minutes left. Thank you. So we made a correlation plot of the area under the curve. You can do that for the whole data set, but you can do that per protein. So we do it per protein. Each dot in these plots is a protein for the 3 different test sets. And then the horizontal axis is the sequence identity of each of those proteins to the closest protein in the training data of the method. But it's different for the 2 methods, the different training data. But now you can see whether a protein that looks like something in the training data gets better predictions. How can you see that? Well, remote homologs are on the left, close homologs are on the right. So if your method performs better on the right than on the left, somehow there's data leakage between your training data and the test set, somehow. If you still figure out how. And then you can calculate the correlation coefficients. And then for second site, that other method that was published a week before we submitted, you have significant correlations for both of these. Well, for our method, graphically, the difference doesn't look that big, but these correlations for our method are not significant. For this very small method, it's only 25 proteins. It's a slightly different story. Probably a small number artifact there. So this is how you can find whether there is any data leakage. Then you still have to figure out why. We don't know. We want to discuss this with the authors. We haven't had the opportunity yet. Now, that's some more details. So One of the issues is that this ZK448 dataset has been reused by many, many groups over many years. And that means that you effectively have a meta training cycle from people reading my paper or somebody else's paper on this data set, making design decisions on how to build their method, testing it on the same data set, publishing the paper, somebody else reads that paper, makes other better design decisions. So as a community, you're learning on this one data set. So it's no longer, after one cycle actually, it's no longer external, new, unseen data. It's part of your meta training loop. So this probably explains why Second Sight was performing better on that benchmark data set, because effectively being meta trained on. This is just a selection of different methods over the years that we're using the same benchmark data set. So bottom line of that is, if you publish something like this, always, always, always include a test set that nobody, including yourself, has ever used before. So you can use a test set only once. Then you have to find new data. Okay, so we have a web server. And the final thing I want to mention is also looking to the future. Better models need better data. And one of the things is that we don't have metadata for protein interactions. Protein interactions in the lab depend on pH, salt, different co-solvents, protein concentrations, other proteins, any number of chemical factors. So we were developing a tool to annotate, to help annotate these details from published papers. So you build like a gold standard data set on which to benchmark protein, not protein, natural language models that people use to extract this data automatically from papers. But if it's a model, you need to test this, you need to test data, and it has to be accurate. So this is ongoing work with Niki Athanasiadou from Rockefeller University. Visiting her in 2 weeks, very excited about that. I do really cool science. Science happens between people. Eric already said that. The developments are breathtakingly fast, but one of the things that doesn't change is the people. Some of us in EMBNet have been in research in bioinformatics for decades, and the method changed, the data changed, the challenges change, society changes, but we're still a scientific community. And the people in the community change, but the community is still there. Okay, so main points. I don't want to spend too much time on that. I'll just give you this as a visual input for coming up with nice questions. Thank you very much for your attention.